In today's video, we shall discuss about the detection and assay of fermentation products. But before that, do subscribe to my channel and let me know your opinions and suggestions in the comment box. Fermented products are those products whose production involves the action of microorganisms or enzymes which cause desirable biochemical changes. They are the outcome of fermentation process. Fermentation products can be broadly divided into two categories: high volume, low value products, or low volume, high value products. The extraction and purification of fermentation products may be difficult and costly. Ideally, one is trying to obtain a high-quality product as quickly as possible at an efficient recovery rate using minimum plan investment operated at minimal costs. The choice of recovery process is based on the following criteria: the intended use of the product, the magnitude of biohazard of the product or broth, the concentration of the product in the fermentation broth, the impurities in the fermenter broth, the minimal acceptable standard of purity, the intracellular or extracellular location of the product, the physical and chemical properties of the desired product, and finally, the marketable price for the product. So, what is an assay? An assay is an investigative procedure for qualitatively accessing or quantitatively measuring the presence or amount or the functional activity of target entity, the analyte. Before assaying, microorganisms strain have to be selected, screened to know if it's the right microorganisms that are potentially valuable in producing a commercially useful product. Three main categories of assay exist. They include physical-chemical assay, biological assay, and chromatographic assay. Physical and chemical assays. Physical and chemical assays are essential for analyzing fermentation products in various industries, including biotechnology, food and beverage production, pharmaceuticals, and more. These assays help monitor and control the fermentation process, ensure product quality, and optimize production efficiency. Types of physical and chemical assays includes titration and gravimetric assay, turbidity analysis and cell yield determination assay, spectrophotometric assay, and other techniques such as infrared spectroscopy, nuclear magnetic resonance, and mass spectroscopy, etc. Titration and gravimetric assay. Titrometric analysis is a type of quantitative analysis in which we can measure the amount of an unknown compound using its volume. A second solution or reagent for the determination of the volume of the unknown compound present in a sample. Through the determination of the volume of the unknown, we can determine the concentration of that compound in the sample. Whereas gravimetric analysis is a technique that comes under quantitative analysis where we can determine the weight of an unknown compound in a sample. In this method, the major step is the precipitation reactions, which leads to the separation of the desired compound from a given sample. The figure here represents the titration and gravimetric assay. Turbidity analysis and cell yield determination assay. Cell yield determination assay. The determination of cell in the given fermentation broth is done by cell yield determination assay if the cells are only insoluble products present in the medium and grow as pellets. Cells along with the medium are centrifuged in graduated tubes and the volume of sedimented cells measured in cubic centimeters. Turbidity analysis. Turbidity is another technique to study the cells in the fermentation broth as product. This technique is useful for the organisms growing throughout the medium. The cells suspended in their growth medium is diluted to a turbidity range that measures quantitatively as optical density with nephelometer or colorimeter. By the deflection of light that is caused by the microbial cells when suspended in the light path of these instruments. Spectrophotometric assay. Spectrophotometric assays utilizes various types of spectrophotometers to measure amount of visible light absorbed by colored solution at specific wavelength, amount of ultraviolet light absorbed by compound, or the intensity of the fluorescence emitted by the compound when exposed to ultraviolet light. The use of spectrophotometric analysis to assay a previously unknown fermentation product is more difficult because the pure compound may not be available for use as a reference standard in preparing standard curve. Spectrophotometric assays are now being used widely for the detection and assay of fermentation products. Other techniques. Apart from the mentioned techniques, if one want to get structure or the chemical composition and bond formation between each atom and given compound, then techniques like infrared spectroscopy, nuclear magnetic resonance, and mass spectroscopy is useful. For such techniques, compound must be in pure form. These techniques are costly but can provide valuable information of the product. Biological assays. For biological assay, sensitive test organism or enzyme is required. The product tested against sensitive organism may depress or stimulates the growth of the sensitive test organisms. On solid media, decrease or increase in growth of sensitive organism can project as zone of exhibition or inhibition. In liquid media, growth of test organism is measured as turbidity of the media. If biological assay employs enzyme, then quality and quantity of the product measured as increase in the enzymatic activity. For categories, biological assays are diffusion assay, turbidimetric assay, metabolic response assay, and enzymatic assay. Diffusion assay. Diffusion assays carried out on solid media, usually an agar medium, which is suitable for the growth of the test organisms. The compound to be tested is allowed to diffuse through the medium in a radial fashion so that the adjacent growth of the test organism is depressed, as in the case of an antibiotic, or stimulated, as in the case of growth factor. The diameter of zone of inhibition or zone of exhibition reflects the concentration of the compound used for assay, and it compared with similar zones produced by various unknown concentrations of standard or reference compound. There are two types of diffusion assays: cylinder method and paper disc method. Turbidimetric assay. Turbidimetric assay are performed in liquid medium. The compound used for assay and growth rate or total growth of the organism used for test is measured in terms of turbidity of the medium. Growth may increase or decreases depend on the compound used for test. Turbidity measured visually or spectrophotometrically or as optical densities or absorbance. The optical densities plotted against the concentration of standard to obtain a standard curve. Metabolic response assay. Metabolic response assays are similar to turbidimetric assay except that, instead of measuring the effect of the fermentation product on the rate of growth or the total growth of the test organism, we measure the effect on some metabolic reaction that the test organism carries out during growth. Organism may evolve carbon dioxide gas or uptake oxygen gas during particular metabolic process when certain compound is present. Thus, the compound types, its concentration, and its uptake measured indirectly by measuring the amount of gas evolved by the organism under standard conditions. All types of microbial response may be placed either of the following categories. Growth response. Most microbiological procedures that are used for analysis depend upon the growth response of the microorganism to the environment. If the organism responds to the substance being assayed with increased growth, it's positive. But if the organism fails to grow in the presence of a particular material, it's negative. Metabolic response. In addition to the growth response of an organism to a particular substance, there may be a metabolic reaction to the material. Certain organisms produce metabolic products which are measurable, which may have a change in some function that can be measured. Enzymatic assay. Enzymatic assays are highly specific, and they will quantitatively detect minute amount of fermentation product, as well as differentiate biologically active and inactive compound. An enzyme preparation incubated with a sample of culture broth caused some enzyme-mediated change in the fermentation product, such as partial decomposition with consequent formation of a measurable product. There are certain limitations for bioassays. It is more difficult to perform this bioassay. It provides greater error. It is less reproducible. It requires specific organism. Moreover, test organisms should have following characteristics. It should be genetically stable. It should respond in a graded manner only to test compound and not to other materials that may be present in the solution for assay. It should grow relatively quickly on simple media. It should not be a pathogen. The cell should not clump for turbidimetric assay or swarm across the agar for diffusion assay. It should, if possible, aerobic or facultative aerobic. It should grow well at pH that does not affect the stability or toxicity of the material. Chromatographic assays. A chromatographic assay for fermentation products typically involves the use of chromatography techniques to separate and quantify the compounds produced during a fermentation process. High-performance liquid chromatography, HPLC, and gas chromatography, GC, are commonly used methods for this purpose. Chromatographic partition assay. Paper and thin layer chromatography are forms of partition chromatography. The solute or sample partitioned between a stationary phase, such as paper or silica gel of thin layer plates, and a mobile phase consisting of a mixture of solvents as these solvents migrate across the paper or silica gel layer. Paper chromatography is applied for water-soluble compounds, while thin layer chromatography is used for hydrophobic compounds. High-performance liquid chromatography, HPLC. HPLC is an analytical technique used to separate, identify, or quantify each component in a mixture. The mixture is separated using the basic principle of column chromatography and then identified and quantified by spectroscopy. It is an improved form of column liquid chromatography. Instead of a solvent being allowed to drip through a column under gravity, it is forced through under high pressures of up to 400 atmospheric pressure. Gas chromatography assay. The gas chromatograph utilizes a form of partition chromatography to volatilize and separate certain types of fermentation products. On injection into the gas chromatograph, the fermentation product converted to gaseous state, and in this state, it pushed by a stream of inert gas through a partitioning column. Compounds separated during passage from this column detected electronically with the help of detector system. If product is not volatile, it is made volatile with the help of chemical-like esters. Significance of detection and assay of fermentation products. The detection and assay of fermentation products have wide-ranging significance across various industries and scientific disciplines. It helps ensure efficient and safe production processes, maintains product quality, contributes to research and development, and plays a critical role in environmental and healthcare applications. Process monitoring and optimization. Quality control. Bioprocess development. Yield estimation. Fermentation control and regulation. Environmental monitoring. Microbial strain development. Waste management. Environmental impact assessment. To conclude, the detection and assay of fermentation products are integral processes with far-reaching implications in diverse fields. From ensuring the quality and efficiency of industrial fermentation processes to advancing biotechnological research, improving healthcare diagnostics, and enhancing our understanding of ecological systems. The significance of these assays cannot be overstated. These analytical techniques empower us to monitor, optimize, and control fermentation processes, thereby influencing the production of valuable compounds, the sustainability of bioprocesses, and our ability to address environmental and health-related challenges. Through precise and sensitive measurements, the detection and assay of fermentation products continue to drive innovation and progress in numerous industries and scientific domains, making them essential tools for modern science and technology.